One of the most remarkable things about the brain is its ability to change in response to sensory experience. During development, sensory experience specifies how the neural circuits will wire in order to be able to process information in a sensible manner. And throughout our lives, we learn through changing in the neural circuits inside the brain. The changes inside the brain are largely occurring at synapses. So-called synaptic plasticity changes how synapses function, and that's the topic of today's video. Much of the synaptic plasticity that takes place in the brain is so-called postsynaptic plasticity, where there are changes in the receptor composition of the postsynaptic membrane at excitatory glutamatergic synapses. We've already discussed presynaptic plasticity, and that's a dominant form of plasticity on the millisecond and second time scale in the brain. But long-term forms of plasticity, the types of changes in the brain that might underlie memories that last for our lives, or at least for days, that is thought to largely occur through changes in the postsynaptic density. And that's what we'll focus on today. We'll think about changes that are largely driven by the NMDA receptor that induces synaptic plasticity, and its changes in the AMPA receptor composition that largely underlie the long-term functional differences at synapses. Bliss and Lomo discovered long-term potentiation in the hippocampus. They found that if they stimulated axonal fibers and recorded synaptic potentials in the hippocampus, they could evoke a stable baseline where each time they stimulated some fibers, they would get excitatory postsynaptic potentials, release of glutamate, activating AMPA receptors primarily. They then made a LTP induction protocol. LTP stands for long-term potentiation, and they did this through high-frequency stimulation of the axons that they were stimulating. So they would stimulate axons, say, at 100 hertz for 1 second. That's a very typical protocol for inducing long-term potentiation. So they did that for 1 second, maybe a few times. You might want to repeat that 100 hertz titanic stimulation a few times. And then when they returned to stimulating as they'd been doing here during the baseline period, they found that the response amplitude had dramatically increased. And that's the phenomenon of long-term potentiation. That increase in the PSP amplitude can be very long-lasting. It can last basically for as long as you're able to record. In this case, we've plotted out 2 hours of recordings, but if you're able to record for much longer periods of time, these changes in synaptic efficacy can last for many days when recorded in vivo. So this is a remarkable situation where a brief change in the activity of the neural circuits can induce long-lasting and large changes in the efficacy of synaptic transmission. So how does that occur? Experimentally, you can induce long-term potentiation by a variety of ways. One of the most common ones, the one that I just pointed out, is this high-frequency stimulation of many axons. You might put, for example, field stimulation electrodes where you might stimulate hundreds of axons at the same time. You stimulate these at high frequencies, and during that stimulation period where you stimulate every 10 milliseconds, what's happening postsynaptically is that you get EPSPs coming in, they summate, and often you get action potential firing coming in on top of this. So there's postsynaptic depolarization and often action potential firing of the postsynaptic neurons. And that's one of the most popular ways in which long-term potentiation can be induced. There are also other ways in which you can induce long-term potentiation where you don't change the way that you stimulate the presynaptic cells and you just purely make postsynaptic changes. And one way in which you can do that is in your whole cell recording electrode. If we're recording with a glass electrode the membrane potential of a cell, you can simply inject current, and you can then, of course, depolarize the cell's membrane potential. And so during a baseline period, the membrane potential of the cell might be sitting at, say, minus 70 millivolts. You get EPSPs driven here that are largely driven by the AMPA receptors. You can then depolarize the postsynaptic neuron. That then unblocks the NMDA receptor. You'll remember that there's a voltage-dependent magnesium block of the NMDA receptor. If you now have depolarized the cell and you give the same stimulation to the presynaptic cell, you can now induce long-lasting NMDA currents. And so depolarizing the postsynaptic cell is another way in which you can induce long-term potentiation. And finally, you can also, and again, it's a similar type of thing. You're also injecting currents, but here it's not a long-lasting depolarization, but you can simply inject brief currents to fire action potentials in the postsynaptic neuron. And so if we're recording an EPSP, then we can make brief current injections in the postsynaptic cell to depolarize and fire action potentials here during the actual EPSP that's occurring in the postsynaptic cell. And so there are two ways here in which we can depolarize the cell. One is simply just taking the membrane potential to depolarize potentials, and you may not need any action potential firing. Another way is to give brief current pulses fire action potentials at the same time that an EPSP is arriving in the postsynaptic neuron. And what all these different methodologies for inducing long-term potentiation have in common is that they induce activation of the NMDA receptor. That's the key event for most forms of postsynaptic long-term potentiation. We need to depolarize the cell, get the magnesium out of the NMDA receptor, and what that then allows is for calcium influx to occur. You'll remember that AMPA receptors are not permeable to calcium, but the NMDA receptor, in addition to its interesting voltage dependence, also has calcium permeability. And so when we depolarize the cell, either through high-frequency stimulation, injecting depolarizing current, or firing action potentials in the postsynaptic cell that unblocks the NMDA receptor, glutamate is still bound to the NMDA receptor, and now cytosolic calcium can increase in that postsynaptic area. And that, in turn, causes activation of protein kinases. Many signaling molecules inside cells are sensitive to the cytosolic calcium ion concentration, and at the postsynaptic density, there's a particularly high density of a calcium-dependent protein kinase called calcium calmodulin-dependent protein kinase 2, or CAM kinase 2, and activation of that molecule downstream of calcium increases is thought to be the most important signal that then ultimately leads to an increase in synaptic efficacy. So this is how we envisage the situation at the synapse. In the baseline situation, action potentials will propagate down the axon, will cause release of neurotransmitter glutamate from the presynaptic specialization, and that will largely activate AMPA receptors. We imagine here that under these baseline conditions, the membrane potential of the cell during rest is maybe at around minus 70 millivolts. The NMDA receptor is blocked. There's magnesium that's sitting here at negative potentials, and the released glutamate activates AMPA receptors, and that then gives rise to an excitatory postsynaptic potential. During the LTP induction protocol, we're depolarizing this postsynaptic neuron, either because we're making high-frequency stimulation of many different axons, and so the whole postsynaptic neuron is seeing a lot of glutamate across its entire dendritic arborization and thus depolarized, or because experimentally we've depolarized the membrane potential to, say, 0 millivolts, which is a typical manipulation to do. That then gets the magnesium out of the NMDA receptor, so the magnesium is now gone. The NMDA receptor can then contribute when glutamate is bound to the NMDA receptor. we're at these depolarized potentials, and now calcium can flow in through the NMDA receptor. And as mentioned, there's a high density of calcium calmodulin dependent protein kinase 2 in this postsynaptic density region. And this kinase adds phosphate groups to a variety of different targets. One target is the AMPA receptor itself, so it can add a phosphate group onto the AMPA receptor, and that seems to change the conductance of the AMPA receptor and make it larger, and that in itself might contribute to enhancing the amplitude of the postsynaptic potentials. However, what turns out to be the more important contribution to postsynaptic long-term plasticity is the recruitment of additional AMPA receptors onto the postsynaptic area. And so AMPA receptors are actually floating around on the plasma membrane at many locations across the dendritic arborization, and they're mobile. They diffuse in the two-dimensional plane of the plasma membrane. And one of the things that seems to happen during long-term potentiation is the ability of the postsynaptic density to bind AMPA receptors and hold them tightly locked in place here at the postsynaptic density. And so one of the major features that CAM kinase 2 is thought to do is to make more slots, as it were, binding partners here where AMPA receptors can bind tightly at this postsynaptic area here, and we then simply recruit more AMPA receptors into the postsynaptic area. And so whereas before we might have had 10 AMPA receptors sitting in the postsynaptic density, after long-term potentiation we might now have 20 AMPA receptors that have been driven in and bound here to the synapse through the activation of these signaling cascades downstream of phosphorylation via CAM kinase 2. The net result is that we have more AMPA receptors when we now return to the situation where we simply measure the potential. So we've done the LTP induction protocol, we depolarized here, but only temporarily while we were inducing synaptic plasticity. We then return to the same conditions as we had in our baseline recording period, but now the postsynaptic potentials are much larger because we simply have more AMPA conductance that's present postsynaptically. Now, if long-term potentiation were the only thing that went on in the brain, synapses would always get larger and larger. And that clearly isn't feasible for all the synapses in the brain simply to grow. So it turns out that luckily there's a converse process called long-term depression that also takes place in the brain. And through the process of long-term depression, the baseline synaptic efficacy, the postsynaptic potential amplitude, can be made to grow smaller through a so-called long-term depression induction protocol. And so we start off with PSPs that are relatively large in amplitude. We do the long-term depression induction protocol, and now in a stable way across many hours or days, we can decrease the amplitude of these excitatory postsynaptic potentials. In order to induce long-term depression, typically many axons are stimulated at an intermediate frequency of, say, 1 Hertz. And so the baseline EPSPs might be stimulated with an inter-stimulus interval of 10 seconds. And so we record baseline EPSPs. Then there's a period of stimulating at 1 Hertz for many minutes, maybe for 5 minutes or 10 minutes. There's a constant 1 Hertz stimulation of the same axons, and then we return to stimulating every 10 seconds, and then it turns out that the EPSP amplitude has decreased in many different synapses across the brain. So that's one way in which we can induce long-term depression, is through 1 Hertz stimulation of many axons simultaneously. Another way of doing it is without changing the interstimulus interval of the presynaptic axons, but again just making postsynaptic manipulations where one can inject a weak depolarizing current through the whole cell recording pipette. So before, for long-term potentiation, we might have been depolarizing the cell to 0 millivolts and then strongly unblocking the NMDA receptor. In the induction of long-term depression, you might depolarize the cell, say, to minus 50 millivolts. A sort of an intermediate situation where one might expect some degree of unblock of the NMDA receptors, but nowhere near as big an unblock of the NMDA receptor as you'd get at 0 millivolts. And so the NMDA receptors are presumably partially activated, and indeed many forms of long-term depression require NMDA receptor activation, but it's at a smaller scale than for inducing long-term potentiation. Downstream of NMDA receptor activation, it's a little bit unclear what happens. Calcium rises, or at least cytosolic calcium is required postsynaptically, but it's also possible that the NMDA receptor has additional signaling capabilities, and it may be that protein-protein interactions between NMDA receptors and other signaling proteins in the postsynaptic density are actually the ones that affect the expression of long-term depression. It's also known that the activation of protein phosphatases is essential for long-term depression, and in particular, the protein phosphatase calcineurin is essential for long-term depression. And phosphatases, they do the opposite of kinases. So a kinase adds a phosphate group to a protein. A phosphatase does exactly the opposite. It'll take that phosphate away from the protein. And so in some respects, this process looks like the opposite of long-term potentiation. But in fact, there are interesting and important differences between LTP and LTD. They're not directly reversals of each other. In terms of what happens after the NMDA receptors have been partially activated and the protein phosphatases have been activated, presumably there's a dephosphorylation event that occurs, and in particular, it seems that, in part at least, directly dephosphorylating AMPA receptors appears to be a key aspect to long-term depression. After that, the AMPA receptors are simply removed from the synapse, at least in part. And so we start off with a situation where we might have many AMPA receptors postsynaptically, and after the long-term depression protocol, we end up with fewer receptors postsynaptically, and so we have less conductance, and therefore we get smaller postsynaptic potentials. So that's the basis of long-term depression. There's another form of synaptic plasticity that probably relates closely to NMDA receptor-dependent long-term potentiation and NMDA receptor-dependent long-term depression that we've been talking about. But this is a mechanism that has at least a more physiological bend to it, in the sense that it relies upon action potential firing and the precise timing of those action potentials. So now we're not talking about long-lasting depolarizations or high-frequency stimulation manipulations that experimentally, of course, can be carried out in a dish, but we have no idea whether they actually take place normally under physiological conditions inside the brain. Spike timing dependent plasticity relies on the precise temporal relationship of an action potential in a presynaptic neuron and an action potential in a postsynaptic neuron. And of course, in the brain, we know that both presynaptic and postsynaptic cells fire action potentials. And there was an interesting discovery by Henry Markram and Bert Sackmann that the precise timing of action potentials in the presynaptic and postsynaptic neurons can make a major impact upon the synaptic efficacy. In particular, what they found was that if we imagine a synaptically connected pair of neurons where we have an action potential in a presynaptic neuron, that then releases glutamate onto the postsynaptic cell, causing, of course, a unitary excitatory postsynaptic potential. They then began to inject currents into the postsynaptic cell at the same time that there was also a synaptic input. So we have the presynaptic action potential that releases glutamate onto the postsynaptic cell as before, causing the postsynaptic potential. But now, in addition to that EPSP, there's also a current that's being injected into the postsynaptic cell, and that then makes it fire an action potential, riding on top of the postsynaptic potential. When one then goes back and looks what happens at the EPSP when we just stimulate the presynaptic cell, now the EPSP has grown. It's important to note that typically this process, this pairing process of presynaptic followed by postsynaptic action potential, is typically repeated many times, maybe 100 times, in order to generate a robust increase in the efficacy of the connection between the presynaptic and the postsynaptic neuron. And so we go from a small baseline EPSP to a large EPSP after the spike timing dependent plasticity protocol has been run, and in particular it's with a delay in the postsynaptic spike relative to the presynaptic spike. At these so-called positive delta T intervals, that causes an increase in the efficacy of the EPSP. Remarkably, if you move the timing of the action potential to be just 10 milliseconds before the presynaptic action potential, then the plasticity reverses sign. And so here again, we're looking at a unitary synaptic connection, action potential in the presynaptic cell, postsynaptic potential in the postsynaptic cell. An action potential can then be evoked in the postsynaptic neuron by injecting current into that postsynaptic cell, firing an action potential, and you can do that at a time that precedes the action potential in the presynaptic cell. So the presynaptic cell evokes an EPSP, just like it did before, but now that EPSP is preceded by an action potential here during the spike timing dependent plasticity induction period, where again you might pair 100 times the action potential and the EPSP, but now the timing is very different to before. Here the action potential was riding on top of the EPSP, now the action potential precedes the glutamatergic input onto the cell. And what is found is that the EPSP now decreases in amplitude, and so we now have a decrease in the efficacy of the EPSP when we've reversed the timing of the pre and the postsynaptic cells. And so this is at so-called negative delta T intervals where the postsynaptic cell fires before the presynaptic cell. And by measuring what types of plasticity happen when you vary this time interval between presynaptic and postsynaptic spikes, you can build up a timing of the plasticity of the change in the EPSP amplitude. So that's what's plotted here on the y-axis, where positive values here indicate increased, enhanced EPSP size, and the negative values are decreased amplitudes in the EPSP. And here on the x-axis, we're looking at the difference in the timing between the postsynaptic spike and the presynaptic spike. At positive times, that's when the postsynaptic cell fires after the presynaptic cell, and that then gives rise to enhanced synaptic plasticity. And when we have the timing the other way around, the postsynaptic cell fires first, that gives rise to decreases in the EPSP. And this temporal relationship to synaptic plasticity, the so-called spike timing dependent plasticity, STDP, has been now found at a large variety of synapses in the brain, and so it appears to be an interesting and perhaps general rule. What's remarkable is really the short timescale here, where we're thinking about 10, 20 milliseconds and maybe 50 to 100 milliseconds on this side here, where on this millisecond timescale, the order of the action potential firing makes a very big difference to the types of plasticity that are involved. And to some extent, this makes sense. And so this EPSP didn't just come at a random time, it came at a time where the neuron actually fired action potentials. And so one might then think that this input to the cell was useful and something that should be kept and maybe even strengthened. And that's what happens here in spike timing dependent plasticity. Apparently, useful inputs that contribute to firing the postsynaptic cell get strengthened. On the other hand, in this scenario, the action potential occurred before the glutamate was even released from the presynaptic neuron, Clearly, the firing of the postsynaptic cell has nothing to do with what the presynaptic cell is doing. And so this input from the presynaptic cell might not be a particularly useful input for that cell. It certainly doesn't contribute to this artificially fired spike that's done under these experimental conditions. And so the cell may think that this synapse is not a particularly helpful one for the function of the neuron. And so it may be the reason for why the depression of that EPSP occurs when the action potentials occur in an uncorrelated way with the presynaptic neuron. Changes in the postsynaptic receptor composition, the number of AMPA receptors in particular that are present at the postsynaptic density, are an important way in which the synapse strength can be regulated between two different neurons. And it's the activity of these neurons, and in particular the depolarization of the postsynaptic cell, concomitant with the release of glutamate from the presynaptic cell, that is typically involved in setting the plasticity and the direction of the plasticity that occurs in these different paradigms. Strong depolarization or action potentials riding directly on top of EPSPs coming in strengthen that synaptic connection. And the strengthening appears to occur, at least in part, through the insertion and recruitment of AMPA receptors that are diffusing on the plasma membrane that then bind onto postsynaptic density proteins and get locked into the postsynaptic density where they can contribute to driving postsynaptic potentials. On the other hand, weak depolarization or action potentials occurring at times that are different from when the synaptic inputs arrive tend to cause a depression of the synaptic inputs. And so there's a strengthening of what appear to be useful inputs, and there's a weakening of inputs that appear to not contribute to the postsynaptic firing.